There's some wonderful work going on in the Parkinson's research field here in Buffalo, and our next speaker is going to talk about exactly that. He was educated at the Nanjing University in Nanjing, China, also at the University of Tennessee. He was a postdoctoral associate at the Rockefeller University in New York City. Currently, he is the director of research in the Pluripotent Stem Cell Facility at the State University of New York at Buffalo. He has earned the Young Investigator Achievement Award, the Visionary Inventor Award, and he has been listed in the top 100 principal investigators at UB. Also authored or co-authored more than 25 articles. Dr. Jian Feng.
So it's a great pleasure to be here. I have been at UB for 17 years. So people in the back can hear me? Good. So the short summary is that our fight against Parkinson is a winnable cause, and we're getting closer. So I'm going to show you what we have done, and hopefully we together can make a big difference. So as most of you have already know by now that movement is actually a function controlled by the brain. So if you think about movement, it's really coordinated contraction and relaxation of muscle, right? And this coordinated contraction and relaxation is all calculated in the brain. And the function of the basal ganglia circuit in the brain is basically responsible for this operation. So the brain is actually a computer. And part of this computing system is to enable us to move. And in order for us to move, we have to have dopamine. So as you know, these dopamine is produced in the dopaminergic neuron. They have their cell body in one part of the brain, and they send very long projections to the target neuron to control these calculations. So when these neuron dies, you will have not enough dopamine to control these calculations, and when that happens, we have Parkinson's. So that's the general idea. And to tell you how special these cells are, so the neurons is a special type of cell, and these cells are extremely unique. So this is actually a study done in the rat, and we have a similar situation. So if you look at in the rat brain, you have a cell. This is the nigodopaminergic neuron. So the cell is, so their cell body is in one part of the area called substantial nigra. This cell has the size of a regular cell, about 10 to 20 micrometers, not very special. What is really special is that this cell extends a very long axon. So this is like a process, it's like a wire that sends to the target area. Once the cell reaches the target area, it sends out massive branches of this axon, so it can cover a huge area of the target area. So this really makes the cell really very unique and very vulnerable, in that if you just do a simple calculation, about over 99% of the volume of the cell is actually in these axon arbors. So imagine that this is a huge tree with lots and lots of branches. And that really makes the cell very valuable for the calculation, because it can control many, many neurons. And at the same time, it also makes it highly vulnerable, particularly for humans. Because the unique situation for these cells is that if you add up the length of these axon branches in the rat, for a single cell, these axon branches totals about 45 centimeters. So this is like about 2 feet roughly. So for human, because we have a much larger brain, what happens is that we still have the same wiring plan as the rat or the mouse. So all the mammals have the same wiring plan. So that entails that we have a much bigger projection field. So what is 45 centimeters for the single cell in the rat now becomes 4.6 meters. So this is like about 5 yards. So this is really, really huge. And the ramification for that is that these dopamine neurons are actually the largest cell in your body. Because if you calculate the volume of this cell, it's actually about 4 times the size of a human egg. The human egg is generally thought to be the largest cell in human. So with this, we have an intrinsic appreciation for the uniqueness of these cells and why it is so difficult to study Parkinson's in animal models. So we need to study human dopamine neurons. It was not possible about 10 years before, and there was remarkable discoveries within the past decade that enable us to study these cells. And we can now generate these cells, we can study them. So if you look at what we are a collection of about 30 trillion cells. All these 30 trillion cells arrive from one cell, so that's the fertilized egg. So every one of us has our first cell. The cell divide and divide again. And roughly about at about day 5 in our life, we have this ball of cell called blastocyst. And inside the blastocyst, you have the inner cell mass. These cells are what we call pluripotent because they give rise to all the cells in the body. And the cells over here, they give rise to placenta, umbilical cord, and those other supporting stuff. So basically, if you think about this, all life forms are actually a DNA-based software system. And the way to read this software is actually at this precise point around day 5, when you can generate these cells that can give rise to all the cells in the body. So now we have ability to make cells that are very similar to cells in the inner cell mass of the blastocyst. So basically, if you really sit back and then look at us in a very dispassionate way, we are a collection of cells, and in each of these cells, the sequence of DNA is the same, the ATGCs, right? And basically, the cell is able to read these instructions and then generate different type of cells, and these different type cells interact with each other. They allow very different things, such as breathing, such as competition for supporting movement, right? So dopamine is one of the subsystems, basically. It drives locomotion. And with this realization, we know that we can, it is a finite problem. It is a finite problem because our brain is a finite system, and it only takes about 20 watts to run the brain. And we actually, if you look at the man-made system in parallel, it's actually truly remarkable that we can actually make robots that move more or less like human beings. So we're trying to understand what exactly is going on in movement, how we can actually understand the control of movement, and then hopefully we can translate that to understanding of Parkinson's disease and generate therapies. So to tell the long story short, we understand now that we need to have these human dopaminergic neurons. So there's a remarkable discovery first in 1998. In 1998, Jimmy Thompson in Wisconsin found that we can actually generate human embryonic stem cells. So basically, human embryonic stem cell can be generated from these cells inside the blastocyst. But in doing that, you have to destroy the embryo, and it has huge ethical issues. And then in 2007, there was a truly remarkable discovery by Shinya Yamanaka in Japan. What he found is that you do not have to destroy the embryo now. What you can do is that you can actually get any somatic cell, so any cell in your body, you can actually convert that into a stem cell. These are called induced pluripotent stem cell. So what you need to do is to put 4 genes into these somatic cells. For example, your skin cell, you put these 4 genes, they try to rewire how the cell is read. So imagine your cell is a computer, right? Your computer is read in many, many different ways to generate different type of cell. You put in these 4 genes, you can hijack the system so that now the cell is read differently. So the hard drive is read differently. So you can actually generate a stem cell that is for all practical purpose, just like the embryonic stem cell. So you can generate the embryonic stem cell without the need for embryo. And with this, we think that it's really truly remarkable because that allows us to make human dopaminergic neurons. So what we can now do is that you can collect, for example, skin cells. You can do a simple skin biopsy, or you can simply just collect cells from urine. So whenever we urinate, we flush out lots of cells, and then we can actually collect these cells in a totally non-invasive way, no pain involved. You can then convert these cells into the iPS cells. These are called induced pluripotent stem cells. So basically, I said it's just like the embryonic stem cell without the need for embryo. Here you have hundreds of these cells. They form a colony. So once you have these cells, remember that these are the cells that generate everything in the body. So you can actually make in a dish anything you want to make. And in our purpose, we want to make the nigroDopaminergic neuron. So you can see that we can actually, I don't know if it's very clear on the screen, but you can actually make these neurons, and a lot of these neurons are actually dopamine neurons. So this TH, this red thing, is actually a marker for dopaminergic neurons. So we can make tons of these neurons. So once we make these neurons, we can use that for a variety of studies. So the first thing, obviously, that came to mind is a cell replacement study. We can transplant these cells into the patient brain to see whether they can be used as a therapy. The second is that we can use these neurons to screen for new drugs. Now, so I will talk more into each of these little aspects. And then the third is that we can actually now use these neurons to study the patient. Basically, it's a surrogate for the patient, is that we can do a variety of things to treat these neurons in a dish. So we can try to understand what it is exactly that someone gets Parkinson's, other people do not get Parkinson's, and how we can deal with that. And then we also, with these neurons, because they're very similar to the dopamine neuron inside your brain, we can actually try to understand, for example, whether there's some biomarkers, whether there's some expression in certain genes that predispose some of us to Parkinson's. So if we understand those, we can then perhaps generate something like a blood test to predict who's going to get Parkinson's. When we have that, we can treat Parkinson's much early on. Just think about, for example, treating diabetes. If you do not have a blood test to measure glucose level in the blood, it will be really, really bad. Right? Then you have end-stage diabetes. So those are the four things that people in research are actually trying to do. And to highlight some successful examples. So, for example, in the cell replacement therapy, people initially started out when they use aborted fetuses. They use fetal tissue to do the transplantation. It's mostly done in Europe, with some also done in the United States, starting in the late 1980s. In several cases, in a few cases where it was done successfully, some patient can be off a PD drug for about 20 to 30 years. So for essential purpose, they're sort of cured of their motor symptom. The non-motor symptoms are still there, but as far as the motor symptoms are concerned, when this is done correctly, it is really wonderful. But the problem is that for the vast majority of cases, it is not done correctly because we do not know what makes things to occur in those successful cases. So that's why we need to have a material that we can study without all the ethical concern, without even the sourcing problem. Let's say it's just not viable to have fetal tissue for this purpose. So this is why this discovery is so important, because you can now have unlimited supply of neuron that is essentially just like the neuron inside your brain. So, and also things are starting to turn around, because once pharmaceutical companies are developing those drugs, they were just basically using a lot of screening on cells that are not very similar to the neuron in the brain. Imagine that you can actually just take the cells from your brain and then do it in the dish, and then you can screen things that are much, much better. So I think people, a lot of pharmaceutical companies, some of which we're working with, are actually realizing the potential of this. I think you'll see more and more in that area. So I just want to give you a few examples in our study. So for example, we can generate these neurons from patient, right? From, let's say we convert the skin cell into iPS cells, and then from the iPS cell we can make these neurons. We can actually then put these neurons back into a rat model. So we can actually give the rat a toxin so they can generate a Parkinson-like symptom. And then when you put the human neuron in, we put these neurons in the substance nigra of these rats. You can see that these human neurons, they not only can survive, they can make massive axon arborization. Remember that these cells are truly unique because they sense all these branches. And you can actually, in the rat model, you can see that these neurons can alleviate the locomotor deficit. So we lesion these rats, so they have locomotor problems, and when we put these cells in, they can do better in locomotor test. And there are actually people are gearing up for clinical trials. We try to make these neurons better and better. At a point, it will be ready for clinical trials. Because remember that this is really the replacement for the fetal tissue, because the fetal tissue really gives us the encouragement that this can work when it's done correctly. So in another case, we said that we want to understand how the disease happened. Right? Some of you who have undergone, for example, DBS surgery may have heard about this. So I said that the brain is a computer. For the computer to instruct locomotion, it needs to constantly communicate between neuron to neuron, right? So when neurons talk to each other, we do not exactly understand how they talk to each other, but we know that the information is not repetitive. And in Parkinson's, what happened is that you can actually record a lot of repetitive talking between one neuron to another neuron. When these communications are repetitive, you lose a lot of information. So this kind of oscillation occurs in patients. And remarkably, when we generate neurons in a dish, we can actually also see this. This is actually from people with Parkinson mutations. So there are actually Parkinson's disease that are caused by mutation of single genes. So we can actually see in a dish that these neurons also make this repetitive communication that we thought is linked to Parkinson's. So in normal subjects, in neurons from normal subject, you do not actually see this. And then what we can see is that if we put in the normal Parkinson gene back into these neurons, so you can see that the oscillation is gone. So basically, these oscillations, you can understand it as if they're sort of like stuttering. So when people stutter off, obviously they cannot really communicate very well. So it's somehow linked to the misfiring of these neurons, so they cannot communicate with each other. So the neuronal network will not be able to perform the function. So all these things are very, very good in that it requires we can generate very nice neurons, but the problem is that it takes several months to generate these iPS cells, and then to turn iPS cells into dopamine neuron, it takes another several months. That makes the thing very tedious and very expensive, because during all these several months, you have to culture these cells, and every day you have to change media, and all these media have a lot of growth factors. So we constantly try to find better ways to make dopamine neurons, and then very recently we found a very nice way. We can actually basically convert skin cells directly to dopaminergic neuron in about 10 days. So this is really born out of the realization that this is just a software system. You can hijack the system, try to rewire how the cell read the hard drive, and then you can make cells that you like to make, in this case, dopaminergic neurons. So as I said, that there's tremendous progress. And when we put in these cells into the rat brain with the PD model, you can actually see that these human cells can survive, and they can make synaptic connections with the rat neuron. So as we look at present, what we have for Parkinson is that you have therapies that treat the symptom. These are not disease-modifying therapies. They will not change the progression of the disease. So the disease goes on unabated. Basically, all these drugs that treat locomotor symptoms, they are really trying to mimic the function of dopamine. They either try to mimic the function of dopamine with dopamine agonists, or they try to rescue the deficiency of dopamine, or they try to slow down the degradation of dopamine. Or you can actually hijack the sort of how the brain wiring works, right, by doing DBS to override overactive part of the control module. Looking forward, what we try to achieve is to find disease-modifying therapies. We try to find therapy that can change the course of the disease, and ultimately to try to find a cure. And over there, we have this wonderful system where we can actually generate neurons that are very similar to neurons in your brain. And then with that, we can actually find biomarkers, and we can find all sorts of small molecule drugs like these chemicals, and then we can try to see whether we can put the cells back. So finally, I would like to acknowledge the hard work in people in my lab, and obviously, without your support, we cannot do this. Some of you actually have participated in our study, and I really feel that this is a fight that we have to do together. And by combining our resources in a very meaningful way, I fully believe that the fight against Parkinson is a win.